morning, good afternoon, good evening. Welcome to your day one of this workshop, How Clinical Pharmacology Shapes the Strategy for Clinical Drug Development from New Molecular Entity to Registration. We are really excited to share that we have received an overwhelming response and have over 500 participants from all over the world. Thank you for being here and thank you for giving us this opportunity to share this knowledge. Today's workshop is hosted by Project Dontabhaktuni and SSX, and it is sponsored by PharmaPro Consulting. Some logistics here. All the participants will be muted during this workshop, and you can reach us, or if you have any questions, please use the chat box on your WebEx screen. And we also will be available to answer your questions after the workshop, and you can reach us by email, which is dontabhaktuni.org@gmail.com. I'm your host, Aruna Dontabhaktuni. Something about me. I'm the founder and CEO of PharmaPro Consulting, and also the founder and scientific director of Project Dontabhaktuni. I have about 25 years of experience in the pharmaceutical industry, and prior to having my own company, I was a director at Lilly, and I headed the PK Pharmacometrics group there. In my career, I have supported 60 plus compounds across multiple therapeutic areas, and my expertise range from preclinical to regulatory approval. Now, something about Project Dontabhaktuni. Now, this is a non-profit created to support scientists and students from all over the world in order to help them access to workshops, training programs, along with mentoring programs. This is focused on creating career paths for everybody. And now, how can you support us? You can support us by participating in these workshops and by joining hands with us in becoming part of the mentoring programs and support this knowledge sharing process, and by spreading the word. And thus our motto, which is empowering you to empower others. We are co-hosting this with SSX. SSX is part of the International Society of Study of Xenobiotics, and it shares similar pathway as we are dedicated to, that is for knowledge sharing and training. Many of you who are participating today are part of this, the SSX members, and we really appreciate you being here. This year, prior to this workshop, we had hosted another workshop online, which was on RxODE tools. And last year, a few of us had visited India and China and hosted multiple workshops. And so far, we have reached over 800 participants, which we are really proud of. Now, without further ado, before I move on, I did want to mention that our next workshop is going to be on R for pharmacometrics, which is going to be led by Rajan from Manipal College of Pharmaceutical Sciences. And without further ado, I would like to invite our speaker for today, Partha Nandi. Thank you, Partha, for taking your time and participating in this workshop and leading us through this workshop. So something about Partha. Dr. Nandi received his bachelor's and master's degree from Jadavpur University in India. Then he went on to get his PhD in pharmaceutical and computational sciences from University of South California. Following his graduation, he did his postdoctoral research in University of California, Los Angeles. Partha joined J&J in 2006 as an associate director in advanced PKPD modeling and simulation group. Soon after, he was assigned the responsibility of PKPD advanced modeling and simulation group in the US. In 2007, Partha was appointed as a director within the group, and in 2013, Partha joined the MBDD group as the head of biologicals modeling while continuing to manage the US-based pharmaceutical modeling group. In 2014, Partha was appointed to the role of regional head within clinical pharmacology, and in 2015, Partha was appointed as the group leader for cardiovascular metabolism and neurosciences, where he's responsible for all clinical pharmacology and pharmacometrics deliverables. Since joining the organization, Partha has made significant contribution to several programs in anti-infectives, oncology, cardiovascular metabolism, and neuroscience disease areas. Partha has authored and co-authored more than 50 manuscripts, 1 book chapters, and numerous abstracts. Thank you, Partha. Thank you for being here, and thank you for sharing your knowledge with us here. So now we can hand over the presentation to Partha.
So
Sure.
thank you. Thanks to the organizer of this webinar session and the workshop. Thanks for inviting me to share my experience in clinical pharmacology, which I'm involved with for several years now. What I'm going to do today is just give you a taste of what clinical pharmacology is and what it does in the field of drug development and why it is such an important discipline that shapes the scope of development and also helps write the right kind of label, which is the most important contribution of ours. I'm going to touch upon many different topics, and as you will see, each of these topics merits a full workshop, a day plus long workshops each. So this will be just I'll provide you with a orientation to the topics with some reading material, which you can go back, read up some of these references, and then through the workshop material, the exercises, I hope that you will have a better understanding and knowledge of some of the topics that we are going to touch upon today. So as a disclaimer, this presentation and information are not the opinion of my past or present employers. It's the opinion of myself as a presenter today. Now, when we think of drug development and think of tablets or capsules, mostly solid dosage forms or even injectables, etc., the predominant formulation that we see in the market are tablets for numerous reasons, as you all are very well aware of. But not all drugs or all tablets are made equal. Along the way, we have to tailor the tablet and its profile based on the quality of the pharmaceutically active ingredient, the disease, the patient population that this drug will be used in, etc., etc. And this is a multifaceted approach that requires people from different walks of life with different skill sets. And we, as clinical pharmacologists, harness all this knowledge and put together a comprehensive program that we then execute in order to get the drug to the market. I'll start with sharing with you where ClinPharm engages along this drug development continuum. It starts with the discovery and screening. We then go to the preclinical. We then proceed into what we call early clinical development, starting with phase 1 studies, proof of concept in phase 2, 3, and then in phase 3, we have the registrational studies leading to NDA. And our work doesn't stop there. We continue into post-marketing, where we look at special population, we look at new indications in the life cycle management program, and we extend the product to be used in the pediatric population or geriatric population as the demand rises. So if you can imagine that clinical pharmacology is part and parcel of this entire drug development process. Although our responsibilities and what we do for the program evolves over time along this continuum, but as you will, I hope to leave with you this message that we are always there as working with our clinical colleagues and regulatory colleagues to make the product more acceptable to the patients. So in short, what do we do at these 3 different distinct stages of development? So starting with early stage, we are mostly answering or asking the following questions, like which candidates are most likely to succeed, what biomarker should we capture, and what dose range is needed to compete with the leaders in the drug class. Now, if we are the first in class, it's good for us, but then we have to explore the safety and tolerability of these drugs and off-target engagement of our molecule much more than one would do if we are working within a certain class that has been studied or explored extensively previous to us. In development stage, we are looking at what indications are most promising, what is the optimal dose, what is our drug product profile relative to competitors. Again, the question will be slightly different. If we are first in class, the question might be, okay, how do you have a compound that is best in class and remains best in class in spite of competition that is either following you or you perceive to be a competition down the road. Approval and post-approval stage, the questions are slightly different. Here we are more focusing on differentiation in the marketplace. Sometimes within the same disease area, there are multiple drugs of different mechanisms. So we try to differentiate between these different mechanisms to understand which one delivers a better therapeutic index. How do we facilitate market access? This is more a question that is in the minds of every pharma company out there. And what are the other indications? Is this drug likely to succeed? And this is a complex question which entails not only the work that ClinPharm does, but also what we have learned during the entire course of development. And you all have heard about the word drug repurposing. And there are many examples today where the drug was approved for something else, but it has a bigger market share in a totally different setting. So I'm sorry, I have to close this. So something that we develop as we go along. So if you have time, I would strongly suggest that you read this paper by Morgan. This was a review paper that was written based on their learning from the Pfizer pipeline over several years. And what they showed very clearly is there are two basic reasons why drugs fail. The first one is exposure. By exposure, I mean that we don't know whether we can deliver the drug in the right amount in the right patient population consistently. So there, if you categorize it into two groups, high confidence and low confidence, versus the confidence in pharmacology, sometimes we have drugs that doesn't have a quite distinct pharmacological profile. We somehow know it works. We have seen it in animals or preclinical studies, but we cannot really pinpoint what the pharmacological nature of the product is. So then you could have a low and a high confidence in pharmacology. And what you would see is based on this two simple parameters, exposure and pharmacology, you could see a wide range of response. So when they went back and looked at the phase 2 success rate in these four categories, you can see that where they had a high confidence both in exposure and pharmacology, they had a decent success rate. 57% is a very, very good success rate in phase 2. However, in places where we had low confidence either in exposure or in pharmacology, the results were dismal. And this is a place also where one should consider smart study design that encompasses futility analysis, sample size re-estimation, and adaptive study designs, amongst others. And this way you can explore or do experiments to find where the drug actually works at a minimum cost and to safeguard your resources that way, using the adaptive design approaches. I would also suggest you this paper that was published a couple of years back in AAPS journal, which talks about why drugs typically fail in late development. And in that paper, there are some very good examples, which is built upon some of the very similar concepts of exposure and pharmacology. And you'll see that if we have low confidence in either exposure or in pharmacology, or we have low, we cannot deliver the drug in spite of knowing what the pharmacology is, then the outcome is not going to be as you expect. So we in clinical pharmacology support end-to-end activities, which encompasses a wide range of studies that we do at different stages of development. And what we do here is not just conduct phase 1 studies, which I'm going to cover in some details in subsequent slides, but also we are the ones providing key information about dose and regimen selection, guiding patient selection, and integrating that knowledge. So what we simply call exposure response analysis. Pharmacometrics is an integral part of what we do. It's a toolkit that we visit and revisit along the drug development continuum at many different stages, and some of those are listed below. I'm not going to read all of this. Some of you are aware and very familiar with these tools and how and where it is used, but the way I envision drug development is that we learn through experimentation, and the modeling tools are the way we analyze the results of those experiments to inform our knowledge. The next slide talks about the kind of studies we do and what are the key elements that we focus. So early on, we are mostly concerned about understanding the pharmacology of the drug in the preclinical area to what I would call the translational research space. During the translational research, we also try to understand the safety tolerability of the compound before we explore efficacy. But sometimes we do combined studies, mostly in oncology and few other areas where we cannot study the drug in healthy volunteer population. We utilize patient population over there. Efficacy is something that is explored early too. We fine-tune our formulation again, going back to the two principles. We first understand pharmacology and then understand exposure. And once we are comfortable with both of these two elements, we move into proof of concept, proof of mechanism, name it. There are many different names to the very same thing given. We test the drug out in a population of interest. Sometimes we test multiple indications in one study, which is very common in cancer therapies, and because that helps us to sequence, do the indication sequencing down the road. Other times we would go with the most preferred indication given the pharmacology of the drug or the exposure, or a combination of both. Moving on to the study types, if you just focus on the second row, we do mostly the biopharmaceutics, clinical pharmacology, safety pharmacology type studies, where we evaluate the ADME property of the molecule. And also, as you know, today, safety pharmacology is a big topic. And many of you know the ThoroQT study requirement for submission, but there are other special studies that are often done, like if you are developing a sedative hypnotic driving study, or a driving study is a must. If you are developing a drug that has activity at any of the opioid receptors, usually there is an abuse liability study that's a must. So there are some of these special studies that are done to qualify a drug product. And the other piece that we are very much engaged in with our clinical formulation chemist is fine-tuning the biopharmaceutics property of the product. And just spending a couple of minutes on that. When we start a drug development process, we start with a very crude drug product. Sometimes it's as simple as putting some powder in a bottle and diluting it at the site and dosing it. But that's not how one can bring the product to the market. So, but what we see in the market is a finely designed tablet. So there's an evolution that occurs, sometimes starting around first in human, and maturing before phase 3, sometimes during phase 3, which puts, you'll often see the term pivotal bioequivalence study. That is done when we did the phase 3 with a experimental product, but the market image product is very different. So there we have to show biocompatibility between the product that was studied in phase 3, which is the driver for approval, versus the product that was the company is launching, to show that nothing is different, that it delivers the drug as intended, and as shown in the phase 3 studies. Now, along the way, modeling simulation is a very integral part of this process, and we do different kinds of modeling at different stages to aid the development process. So just very high level, what are the different kinds of clinical pharmacology studies? And here is a representation of the study types. I'm also putting in immunogenicity here For many compounds, if you're working in a large molecule space, you have to assess immunogenicity of the product, which is not required in small molecule. But the rest of it pretty much are the same. Food effect obviously is more relevant for orally administered products. But for large molecule, there are instances where oftentimes you have to do certain drug-drug interaction studies, especially if the drug works through cytokines, and many of the cytokines are known to affect the CYP metabolizing enzymes. Or if you have a molecule that somehow alters the PK of the other co-administered drugs, then the regulators request some drug-drug interaction evaluations. Now, where does all this fit in? This is an example of FDA product label, and you can see that we contribute pretty heavily in the dosage and administration section of the label. We contribute to the contraindication section, dosage form and dose strengths, drug interactions, special population, and finally there is a dedicated section where we describe the clinical pharmacology property of the drug substance and the product. Now, many of you are very familiar with the biopharmaceutics and its associated classification systems. I just want to, take a moment to show this one and describe how we utilize this particular classification system in developing our clinical pharmacology strategy for a product. Some of these, I'm not going to go in details. There are plenty of references in the literature if somebody is interested in reading it up in more details. Also feel free to reach out to me. So the clinical pharmacology studies that we typically do are listed here. I'm not going to describe all of this, but some of the ones that are highlighted here, I'll focus on those today very briefly to give you a flavor of the extent to which we investigate a molecule ability to affect the overall ADME property. Now, the staging of these studies could be very different, and this is a pictorial depiction of what is an ideal timing along the drug development continuum for conducting these studies. This is just a representative slide. It's not the only way to do it. Depending on the question, depending on the drug property, sometimes we move the sequencing of studies around quite a lot. But one thing that I have come to realize over the years is doing the mass balance study with a radioactive molecule early on in the program could help understand the property of the molecule much better and could lead to a very well designed clinical pharmacology strategy. So something to keep in mind, there are different philosophies around it, and there is a different level of reluctance in different parts of the organization in different countries to conduct these studies early on. But this is a very useful study to do early to understand the molecule better. And particularly for drugs that you cannot develop a IV formulation, there are techniques now with microdosing, etc., that can give you an idea of the absolute bioavailability of a compound early on as well. So that's something to keep in mind. Again, I'm going to just highlight some of the other areas where we are heavily engaged and often end up doing conducting studies in this special population, which is what happens to a drug during pregnancy, labor and delivery, nursing mothers, the drugs, pediatric and geriatric use. And often overlooked section is overdose. If you can imagine, the SAD study is a tolerability study. So we push up the dose typically to a maximally tolerated dose in a population. So given that profile, one could inform the overdose situation and what to do. But there are other studies that one could do to understand the overdose. And this is not very common study for majority of the drug development, but is required if you are concerned of some adverse events that could be like threatening or drugs with narrow therapeutic index. So very quickly, the four properties of the drug product and a formulation is very critical for us to characterize. The first is absorption, which very simply we do some early phase 1 studies in healthy volunteers or in patients as deemed feasible to characterize the absorption. Same with distribution. This is usually available through the ADME study. We get a lot of information from the radiolabel study that we do. Metabolism is an area which is quite complex, particularly for small molecules, and the knowledge that we gather from clinical studies are preceded by preclinical experiments, in vitro work, and I'll show you a decision tree on how we arrive at the decision of conducting clinical DDI studies, but it starts with the preclinical exploration of the metabolic property of the molecule. And finally, I want to show you, people ask, so what is an ideal clin pharm drug candidate? So obviously we want a broad therapeutic index. Not that we cannot develop drugs with narrow therapeutic index. It requires a lot more work and very close and careful assessment of the efficacy safety properties. Obviously, we do not want to have a drug that prolong the QT interval because that could lead to torsades du poids risk. But there are drugs, as you know, where QT is manageable, and also not a significant inducer or inhibitor of CYPs and PGP. But there are drugs which we are all very well known, are routinely used in treatment, where we have learned how to manage these risks, but it requires more work, and it's usually an expensive clin pharm program. Again, the ADME studies are done typically with radio-labeled molecules. Recently, there are examples where stable labeled molecules are used, which are the stable isotopes of carbon or fluorine, etc. That could be used as well. That reduces the burden on the patient and on the site, because handling radioactivity requires some special considerations. But these studies, again, are very useful. The other thing that I would encourage everyone to look into is the microdosing. This is where you can give roughly about 100 microcurie of the material, co-injected IV with an oral drug, co-administered, and you can measure both the cold and the hot drug at the same time through different techniques. What it does is, you get a good characterization of the PK. You have a good characterization of the absolute bioavailability, which is a requirement in certain region if you want to get your drug approved there, particularly in Australian markets and sometimes in Canada. But most importantly, you don't have to do a thorough safety evaluation prior to doing a microdose study, because the amount of radioactivity is so low, it doesn't merit doing any preclinical or other kinds of assessments prior to conducting that study. Now, this is the bread and butter of what we do. This is falls under the category of biopharmaceutics. Because the formulation development and site change is a continuous process, and we in clinical pharmacology provide the clinical backing to those efforts. There are special considerations. Every agency out there has their own guidances. What we try to do is to distill out what are the key similarities and differences in each of the region that we are interested in getting the product registered and design a comprehensive BABE study. There is a slight difference in the definition of BABE. That's something is also part of your exercise. And there are some difference in design considerations between a BA, which is a bioavailability, and BE, which is bioequivalent study. And that's something I would encourage you to read up and come back with the exercise at some point. I'm going to share with you the exercises at the end of the presentation. Food effect is very standard. There are guidances around this. Typically, a food effect is done with a high-fat meal, which is over 500 to 600 kilocalorie. But there are drugs which are highly affected by food itself. And any small change in food could alter the pharmacokinetics of the drug. In that case, we do a panel of food effect studies where we look at only carbohydrate food, protein, high fat, and also standard meal, because not everybody is eating high fat, high protein meal every day. And also there is a possibility that there is an interaction between the timing of food and when the drug is given. So then we also look at the time component in these studies. And these studies could get very complex, but that's not usually the case for most compounds. Typically, a food effect study is done with the fasted arm as a control and a high-fat meal as a test. So moving on to the next set of studies, which is renal impairment. This is a decision tree that is typically followed. We try to do a minimal approach as much as possible. So what that minimal approach means is, instead of studying all three categories of mild renally impaired, moderate renally impaired, or severe renally impaired population, we start with the worst case, with the severe, and see at what point we cannot pick up any more difference between the renally impaired group and the normal population. And once we reach that point, we close the study out. This way, not only can we save money, but also we can save on recruitment time and save dosing patients unnecessarily. So it has both a ethical value and also commercial value in doing these minimal designs. Although my strong recommendation will be talk to your regulators in your region if you have thought of conducting a minimal design study, because oftentimes they might come back and say, no, we need the full spectrum studied. So it's always good to have that negotiation based on scientific knowledge. And in all this, keep an eye on the molecule, because if your drug is mostly metabolized with very little renal function, then you probably don't have to do a lot of investment in studying the drug in renal impaired population. However, there is one caveat. Some drugs under certain conditions, actually, it's more of a physiological interaction that happens between metabolism and renal impairment. So some renal impairment could lead to effect on CYP enzymes. And this interaction has been widely studied and published. And it behooves us to explore that to see that, something that our drug doesn't fall victim to that physiological interaction. If it does, then you have to do a much broader exploration. And there are some examples in the literature where you would see drug-drug interaction studies conducted with a CYP3A4 inhibitor in a renally impaired population, because that's where the physiological interaction exists, and that affects the pharmacokinetics of a certain compound. There are numerous of those. You could look into the literature and read those examples. So typically these studies are parallel group design, because you cannot do crossover, as you can imagine. They also are quite small studies. The normal renal function population usually are age, gender, body weight matched, because that way you can compare like to like. And that's another piece that often you'll see we enroll the renal impaired population first, then enroll the normal healthy population just to aid that matching. This is a decision tree for hepatic impairment. Again, there are three child-PU classes, A, B, and C, with different degrees of renal impairment. We follow same strategy mostly. We do minimalistic design, and then if there is a need, either based on the product, based on the disease indication, etc., we will expand the study to all three arms of renally impaired patients, hepatically impaired patients. I'm going to skip that one and go to drug-drug interaction studies. This is the decision tree that we use. It's directly from the FDA guidance document. So all drug-drug interaction exploration starts preclinically in vitro systems and then in vivo systems. And based on criteria that is set, we decide whether or not we would like to do a human drug-drug interaction study. One thing that I want to mention here is there is a lot of literature in this area, but you have to be careful and understand the nuances of these preclinical DDI explorations. There is substrate sensitivity, which you see with the CYP3A4 enzyme in particular. There is also, the interacting drug is not always clean. There are many drugs which affects multiple enzymes in different proportions. So there are not pure inhibitors or pure inducers that we could use in these studies, because one of the limitation of doing human DDI study is that we can use drugs that are approved in that population. So we, and most of these drugs are, they will have a strong effect on one enzyme, but they could also affect other enzymes, and you need to understand that interplay while interpreting the result from these studies. And therefore, you would see a lot of disconnect between different groups claiming different results to be, from very similar experimentation. And that happens because of either some different methodology being applied or slightly different drug being applied, being used. So understanding this is very critical. Typically, the drug-drug interaction studies are done to inform dose adjustment. Sometimes we can, we have to increase the dose or reduce the dose to maintain a therapeutic level. And these studies give us that indication. Typically performed in normal healthy volunteer population, but sometimes there is a need to do these studies in patients. Obviously, for oncology indications, these studies are done typically in the context of the clinical studies. And those of you who are familiar with population PK, it's used very routinely in this context to understand the differences due to, in these drug-drug interaction situation, and PopPK then informs the dose adjustment as well. This is the last and one of the key safety pharmacology study which is done pretty much in every drug development program, particularly if it's a new molecular entity. Starts with in vivo and in vitro and in vivo. So you could start with the HERG assay, which is the human etheragogo gene assay. If you have a signal there, you could also start looking at IKR channel currents. If you see signal, you go into different in vivo models. These are telemeter dogs, anesthetized rabbits, conscious dogs, etc. And each of these models give you slightly different perspective of the QT risk. And if all these models or majority of these models suggest that there is a potential risk of drug-drug of thorough QT in patients at the doses that one needs, and that's an important factor. Like if your therapeutic dose is much lower, then where do you see these preclinical signals? You could avoid doing a thorough QT study. But what often ends up happening is if it's a very large product targeting a much wider population, most companies will go ahead and conduct a thorough QT, which is a pretty expensive study to do. You have to do multiple dose, and the most important there is selecting a supratherapeutic dose. We also need a positive trial to prove the drug, your test compound does or doesn't have a thorough QT liability. Typically moxifloxacin is used, and then there are reference numbers that you have to meet to show the assay sensitivity in that study. So typically 10 milliseconds, moxifloxacin will increase your QT over 10 milliseconds, and you need to have at least that in your study to prove that your test drug, whatever results you got in this study for your test drug is valid. I'm going to spend a few minutes on first in human study because this is one of the key activity that clinical pharmacology, a clinical pharmacologist often engages in. I'm just providing you with the glossary here. You can take a look at this at a later time. Obviously, the starting dose selection is very critical for us. And it takes different shape, and we use different methodology depending on whether the molecule is a high risk, moderate risk, or low risk. So if it's a low risk, it could be low risk because we either understand the risk from predecessor molecules or a class of molecule that has been studied extensively, or truly the risk from this molecule is very low, i.e. we haven't seen any side effect of concern in preclinical models, both rodent and non-rodent. We go through a very intensive preclinical evaluation. We do a lot of PK/PD analysis, and you hear the term PBPK is used extensively at this stage. and we derive, this human starting dose, which often is a challenge because depending on the molecule and its mechanism, we build up safety margin. So our goal typically is to start off with a very small dose that is very, very safe in our first human exposure, and then we build it up. This is outline of how things proceed. There are guidances, regulatory guidances. Our main concern is safety at this point. That's the only focus of these studies. You can go with healthy subjects, patients, mostly in cancer, but sometimes even for other indications, we go into patients because if the drug it doesn't exhibit the kind of safety we want to elicit in a healthy population, but is only evident in a patient population. Those are special cases. There are many different ways of designing this first-in-human studies, and you'll see some very innovative design in cancer area primarily, but now I'm seeing it coming into other therapeutic areas as well. And typically, once we have that, we provide input into the IND process. This is the basic safety factor. If we do go with NOEL, which is the no adverse effect level in preclinical species, and we are going into healthy population, we typically start off with a dose which has a safety factor of greater than 10. And that's according to the FDA guidance. ICH is very similar, but also they are specific to oncology, and there are different approaches to it. MABEL is something that is used in biologics a lot. Also, recently we are experimenting with the PAD approach, which is the pharmacological active dose. And this is an important consideration, particularly if your drug has a cytokine, because cytokine effect could be mitigated by priming. So you start low and you let the system prime, then you can push up the dose very easily. But if you hit somebody, a patient with a high dose, you're going to elicit cytokine storm, which often or could turn out to be fatal in that patient. So if you're certain of the pharmacology, then pharmacologically guided dose escalation is recommended, and perhaps is the only way to do it for drugs that could have a cytokine release system. It could elicit cytokine release. So what do we do to support first in human? Obviously, we have to evaluate the data coming from our toxicology program, our in vitro assays, and also in vivo preclinical assays that are coming in, gene tox, and also some assessment of the expected PK profile of the molecule, and that is done through GastroPlus or SimCip programs. And the FDA guidance and non-clinical safety guidance are a good guide to inform what is absolutely needed. Typically, the duration of preclinical safety assessments are here. It varies between different compounds and also how much risk a sponsor wants to undertake, but there are certain limitations that are enforced by the regulatory bodies and ethics committees that most sponsors have to abide by, but these are the typical numbers. You probably are familiar with allometric scaling. There are different forms of it. We do human dose projection based on allometry for the first in human, and also allometry is used in predicting starting dose in pediatric population from adults, etc. But most importantly, I will again emphasize that one needs to have a very good understanding of the MOA or the pharmacology of the drug. One thing that we often overlook is the off-target effect of drugs, and that's something we could explore starting in vitro and then in vivo in animal experiments. Certain things are not evident in animal experiments, like if your drug has a CNS effect, as a side effect or off-target effect, animal models are not good unless you go to higher animals like non-human primates and beyond. You could see some things in dogs. So you have to be, careful and also constantly challenge the paradigm, like what is it that this molecule is likely to do, and whether the models that I'm using to test the safety, etc., is appropriate or not. Lastly, this is a very quick schematic of how we arrive at first in human dose, starting from our first preclinical experiments. This is a way we calculate our starting dose in healthy subjects. I thought to share this with you to give you a flavor of the number of steps that one goes through. And you can use this if you haven't used it before, to take your example that you have in your research or your workspace and see if you could calculate what the starting dose is. Typically, if it's an old compound, you should be able to find some information in FDA review packages, which are quite readily available, and see how well you are able to assess. This is the human effective dose or equivalent dose calculation tree. There are some. This is from FDA's guidance. They provide the sponsor with some general rule, but it's up to the sponsor to go with their own choices and negotiate with the agency. But this is again a very good starting point, particularly if you're doing your preclinical experiments in say ferrets or guinea pigs, because that's the best animal model you have, or in mini pigs. These conversion factors are a very good starting point. A brief note here on Mabel. Again, I'm not going to go over it in details, but leaving it here, mostly used in case of biologics, but many sponsors and companies are using this now today routinely, because that's also what European regulators are pushing for after a couple of debacles, both in the small and the large molecule area in first in human studies. So this is becoming more and more evident, and is becoming popular also for selecting first in human dose. Finally, I'm not going to go into all the math today. It could get very complex very quickly. And, what we do is to provide estimate of starting dose on different methods, and then have a discussion with the team on their safety, tolerability, expectation. And then we balance that out and come and design the entire dose escalation scheme. These dose escalation schemes are provided as a initial discussion point, but as you can imagine, with healthy volunteer or even in patients first in human study, these dose escalation paradigms are often tailored as we go along. They are constantly altered based on the new kinetic data and the safety tolerability data that comes in from each cohort. Now, one thing before I finish up my talk here today, one of the key changes that happened since late 2018, but fully implemented 2019 onwards in Europe and in US, is use of sentinel cohort in first in human studies. So instead of exposing the entire group of subjects or patients all at once at a given dose, Usually a single patient is given a placebo dose and a single patient is given the active dose at that dose level. And they are followed for a certain period of time to assess any adverse events of concern. And if there are none over a certain period, depending on either the PK exposure or the pharmacology of the molecule or the target, then one could open up the rest of the patients in that cohort to get the dose. And this is done at every cohort level. So you will hear the term sentinel cohort often. That's what it is. And finally, this is my last slide. There are other characterizations here. Oftentimes nowadays we recommend doing thorough QT assessment, because if you have good QT data and there is no signal, one could use this data to seek a thorough QT study waiver, which is a good savings of $5 to $10 million sometimes. And the beauty of these early first-in-human studies is that one explores a pretty wide dose range, which one never does once one has chosen a dose to develop for a given indication. So this is a rich data that could be utilized. We learn a lot about the PK. Obviously, we characterize PK in different matrices. If there is a need to, at this point, to start evaluating the free drug, we do that too, and build that in. Typically, it's done for compounds that bind to alpha acid glycoprotein, which oftentimes is a nonlinear system, because you don't want to have any surprises as you go into a proof of concept study. And we start exploring early biomarkers in humans at this point. But balance needs for details versus flexibility in protocol to account for uncertainty. These studies are quite uncertain. It often goes the way it's planned. So having that agility to adjust and adapt to the changes is very important for success in first-in-human study. With that, I thank you all for your patience, and also thank you to the committee and Project Dontabhaktuni to allow me to speak to you today. Thanks here. Quickly, I'll spend a couple of minutes to orient you to the exercise. These slides and the exercises will be posted on the website at some point, probably later today. What I want you all to do is attempt at least one exercise topic, but you're welcome to attempt all 3 exercise topics. Additional links pertaining to each topic are embedded in the slide deck, but feel free to go over the FDA, EMEA, ICH guidance documents. You'll find a lot of material of value there. The other additional reading could be drugs at FDA. You can do Google search and it will take you to the current page, and you can see different, all compounds that has been approved by FDA. The reviews are posted, and those are very rich information source. So I have one example on drug interaction. The way these are designed is I provided some basic background information for the molecule, and each of these, information that I've provided are critical in some ways in understanding and designing what is asked in the questions. So there is the first topic is on drug-drug interaction. The second topic is on bioavailability and bioequivalence, and the third topic is first-in-human study. So with that, I'll stop and thank you for your attention.
Thank you, Parth. Thank you for this comprehensive overview. And I can see you actually covered practically your entire lifespan of experience in the slide decks. Thank you so much. Really appreciate it.
Thank you. So if there are any questions, I can take few questions if people have time.
So, Rajan, can I request you to look at, I think we have received a couple questions here, and I also do acknowledge that we are 12 minutes over our time, but we want to make sure that you actually get something of value while you're attending this workshop. Rajan?
Yeah, the chat is moving. The
Sure.
question I saw was how this pharmacometric science or this modeling is contributing to the dosage regimen selection. It's a very broad question. That's what we are talking, but that's a question was there. How this modeling is helping to these approaches, modeling approaches in dosing selection, to put in a nutshell.
Right. So, the simple answer to it is we have come a long way since, in past 20 plus years where integrating modeling into clinical pharmacology and decision making has transformed this area. But I also want to remind people of one thing, that modeling is as good as the data one generates. So if you don't have data that one could model, it's very risky endeavor, particularly for compounds or drugs that has uncertain pharmacokinetics and limited or unknown pharmacodynamic property or pharmacology. Modeling helps with interpolation more so than extrapolation. If one is relying on modeling to extrapolate certain information without having good understanding of the science underneath that extrapolation, is very risky. So, giving an example that everyone would resonate with everyone is, if the compound is class 1, typically one could assume that there will be dose linearity and proportionality over a wide range. But making same assumption for a class 2 or a class 4 molecule, BCS class 2 or class 4 molecule, is quite risky. And we also know that sometimes this proportionality doesn't hold for BCS class 3s. If you check metformin, it's a great example. So, I would caution people to be very, very careful when one is extrapolating knowledge. But it's a good starting point. At least it gives us, what I say, a line in the sand to understand what the limits are, what could be, and then tailor the experiment within that, framework and generate the data. I wouldn't propose, or I'm personally, I'm not a great proponent of just relying on modeling without having data support, because it often it causes more harm than good.
Sure. There are a couple questions which are, I would say, simpler here. One is, what is the full form of MABEL?
I think it's in the slide, in the summary. Let me give you here. It's minimally anticipated biological effect level.
I do have to say that we have received quite a few appreciations and thank yous from our participants. At this point, I think we would want to, I do see that there are few additional questions here that we may not have had the chance to get to. But
So, Aruna, what you can do is capture the questions and send it to me. I can work with you or someone to group them into a common class, and then in the next session, we can start with that, provide some answers and go from there, if that helps.
Absolutely. Thank you, Partha. That is what we're going to do is the questions which we have not been able to get to on the day one of this workshop. We will actually cover them on the day two of the workshop. And as we had mentioned earlier, we want all the participants to actually get something of value by attending this workshop. And that is the reason why we are including homework assignments to go with this. Rajan, if I can get the control back, and if I can share my PowerPoint presentation, I can just walk the participants through on what is our next steps. Thank you.
Yes, you can do.
So, all right. Can you see my screen now?
Yes.
Okay. So I just wanted to give everybody an overview of the total participants who have registered for this workshop. As you can see that we have a good spread, and majority of our participants are postgraduates or PhDs and also PharmDs attending this workshop. And we had also requested to get an idea on whether you would like to be mentored or whether you would like to be a mentor while you're attending these workshops. And we do see that there is a very good spread in terms of mentors and mentees. So our next step here would be that we will send you another form to look at whom you can be paired with. So within the participants who are attending these workshops, we're going to create pods that you can work together on. You can choose to work on one or all of the homework assignments. And our promise is that you get supported through doing these homework assignments. What Partha has covered today is a very, very broad overview, but unless you really get your hands in and do this work and read up, you will not be able to gain a lot by attending these workshops, right? So that is our next step. So you will hear from us. Please be on the lookout for an email from us. If you don't by any chance hear from us because of your email filters or however way things are, you're welcome to write to us. And our email address is dontabhaktuni.org@gmail.com. We will share the slide deck, the recordings of today's workshop, and the reference materials along with your homework assignments. And you will also hear from us with regards to which pod you're part of, whom would you be working with. And during this time, while you're working through your homework assignments, if you get stuck at any point, you need any guidance, your first step would be to go to your mentor who's assigned to your pod. And if your mentor is not able to, or it has questions, your mentors will have access to reach to us, and we can direct your questions to Partha if that is needed. And with that, I think we come to the conclusion of today's workshop. Again, none of this would be possible without all the effort that has been put in. We would like to acknowledge Partha for all his time, and thank you for accepting our invitation to come and present and host and lead this workshop today. Ramya, Krishna, Ramesh, They have been really instrumental, who've been sending you emails and staying in communication with you. We also want to acknowledge Tanga for accepting to co-host this workshop with us, and the SSX board for helping us reach you, broader audience, and having you present at these workshops. And our sponsors are PharmaPro Consulting, which is a consulting company, specializes in data management, PK/PD modeling, clinical pharmacology, and regulatory affairs. They have worked with multiple pharma biotech companies in their past 5 years of existence. Again, thank you all for being here, and we look forward to seeing you on day 2 of the workshop, and look forward to staying in communication with you for the next week.